Hey everyone, this is Dylan with BJJ/J Athletic Training. Today's data-driven nutrition section is on branched-chain amino acids, meaning leucine, isoleucine, and valine, the BCAAs. Amino acids are the building blocks of protein. Said another way, all proteins are made of amino acids, and are incredibly popular supplement ingredients. A branched-chain amino acid, or BCAA, refers to the amino acids leucine, isoleucine, and valine. These amino acids are considered essential because your body cannot synthesize them and therefore relies on the diet to obtain them. BCAA and amino acid supplements alike are very often used as alternatives to protein supplementation. The general idea behind BCAA supplementation is that proteins are made of amino acids. So if you ingest amino acids, your body can use them to make new proteins. Supplement companies claim BCAAs to improve muscle mass, endurance, and recovery during and after exercise, promote fat loss and protein sparing, and overall maximize your gains. This review will not compare and contrast protein versus BCAA supplementation, but rather explain the data pertaining to what effects BCAAs can have on our physiology. And remember, I am covering BCAAs as a collective rather than each individual amino acid in the group. With the emerging anti-BCAA supplementation data surfacing in the scientific community, I felt this would be a particularly important topic. Also, keep in mind there are thousands of papers about BCAAs, so I will cover those I find most relevant and important to the page. Some of the earliest research on branched-chain amino acids shows their importance in development, effects on metabolism, and how deficiencies in their metabolism can result in diseases such as maple syrup urine disease, or MSUD. I think the first interesting thing to note here is that MSUD is caused by an enzyme deficiency which prevents the breakdown of BCAAs and results in their elevation in the body. As a physiologic scheme of BCAA metabolism, leucine, isoleucine, and valine are metabolized first in the skeletal muscle. Only a small amount are metabolized in the liver. When in the skeletal muscle, they are converted to their keto forms via the enzyme branched-chain aminotransferase, BCAT. After which, the newly made alpha-keto acids are converted to acetyl-CoA, succinyl-CoA, or acetoacetate via the branched-chain alpha-keto acid dehydrogenase enzyme complex, BCKD. In MSUD, there is a mutation in the BCKD complex, which results in dysregulation of BCAA catabolism, preventing the alpha-keto acid conversion to two carbon molecules. This can cause brain damage, death during pregnancy, glucose handling issues, and severe tissue damage. The discovery of MSUD was one of the first signs that while BCAAs are important to our physiology, their dysregulated metabolism can result in disease. In 1974, there was an interesting article describing the phenotype of obese people. A group from the Yale University School of Medicine showed that obese human subjects have higher levels of BCAAs compared to non-obese individuals. This was some of the first evidence showing moderate alterations in BCAA metabolism, not as severe as MSUD, are associated with disease states. Further, in 2001, two groups showed the mTOR pathway to play important roles in insulin and glucose handling, and discovered amino acids can activate this signaling pathway. In 2002, a group from the University of Vienna Medical School investigated the effects of short-term amino acid elevation on whole-body glucose and insulin handling in human skeletal muscle. They found amino acid elevation to promote insulin resistance and reduce glycogen synthesis in muscle via the inhibition of glucose transport and phosphorylation. In 2009, a group at Duke University Medical Center also showed elevated BCAA levels in humans to correlate with insulin resistance and obesity. In the same study, they found rats fed on a high-fat diet and supplemented with BCAAs reduced their food intake and body weight, yet became equally insulin resistant as their high-fat diet, no BCAA, much larger rat counterparts. They went on to explain that dysregulation of BCAA metabolism leads to insulin resistance, glucose intolerance, and type 2 diabetes. Later, a group found that protein restriction, high-carbohydrate diet improved the metabolic health of humans. When they investigated the mechanisms of how protein restriction improved metabolic health, they found BCAAs to be the culprit, as BCAA restriction improved glucose tolerance and body composition. Elevated plasma levels of BCAAs have also been found to promote an inflammatory response in peripheral mononuclear cells and induce oxidative stress in endothelial cells of mice. This study is interesting because healthy mice were injected with BCAAs, showing that elevation in BCAA levels in the blood can drive disease rather than solely arise as a symptom of disease. Again, this furthers the idea that plasma elevations in BCAAs act as an independent contributor to the development of metabolic disease. The link between elevated BCAA levels and metabolic disease is growing so strong that researchers have dubbed this phenomenon the BCAA signature, and BCAA elevations are used as a biomarker to predict metabolic and cardiovascular disease. A similar study shows chronic elevations of BCAAs in the brain leads to oxidative damage and impairs memory. This was reversed with antioxidant supplementation. So far, we have seen that elevated plasma BCAA levels are associated with and can drive metabolic disease. But how and why do BCAA levels rise and fall in the body? From a physiologic perspective, one can deplete BCAA levels in the blood with fasting and increase their levels in the blood with a meal or BCAA supplementation. A study from the 1980s shows changes in BCAA metabolism in response to exercise in both trained and sedentary rats after 1 hour of treadmill running. They found with exercise the BCAAs and many other amino acids decreased, while glutamine and branched-chain keto acid levels were elevated in the plasma following exercise, again showing that metabolism of BCAAs occurs during exercise. In a separate study, BCAA levels increased with treadmill running when measured in the rat brain and plasma. However, as animals approached exhaustion, BCAA levels quickly decreased. The same effect has been shown in humans undergoing prolonged exercise, measured in plasma, not the brain. Interestingly, BCAA levels have been shown to increase following exercise under normal conditions without intervention, meaning no BCAA ingestion. This elevation was suppressed when insulin was administered post-exercise. Various groups have measured enzyme activity of the complex responsible for BCAA catabolism, the BCKD complex, in response to exercise. One observed increased BCKD activity with intense prolonged exercise, which makes sense given the prior research showing reduced BCAA levels immediately after exhaustive exercise. In general, BCKD is regulated by its kinase, BCKDK. When BCKD is phosphorylated, it becomes inactive and vice versa. So, decreasing the activity of BCKDK will increase BCAA oxidation. There are a number of factors that enhance BCAA oxidation via this mechanism, such as exercise, leucine derivatives, and fatty acid oxidation. To understand how BCAA metabolism becomes dysregulated, a group from Cambridge conducted a large genome-wide association study of 16,596 individuals. They identified 5 regions associated with BCAA metabolism disturbances, with the greatest significance coming from the PPM1K gene. This gene encodes an activator of the BCKD enzyme, which is very important for BCAA catabolism. The authors explain that a genetic variant of the PPM1K correlates with impaired BCKD activation and therefore interferes with BCAA catabolism, resulting in increased BCAA levels in the blood. This study showed that one may be more likely to develop and maintain elevated BCAA levels and develop disease due to their genetics. There is also a very interesting disease called McArdle's disease, MAD, in which patients have a myophosphorylase deficiency and therefore cannot utilize muscle glycogen for energy during exercise. These patients have been used to study BCAA metabolism during exercise extensively. MAD patients produce more ammonia and glutamine, utilize larger amounts of BCAAs, and have faster activation of the BCKD complex compared to controls during exercise. When these patients supplement with BCAAs prior to exercise, their heart rate and plasma ammonia levels increase and performance decreases rapidly, showing increased reliance on BCAAs for energy production is not conducive to healthy exercise. It has been shown that enhanced BCAA metabolism drains important reaction intermediates and impairs aerobic exercise and the use of glucose and fatty acids. From this, we can now say that BCAA levels can rise and fall for a couple reasons. Levels may elevate due to your body absorbing a meal or BCAA supplement you have ingested. They're released from muscle or other organs via protein catabolism because they are not being degraded properly due to a lack of BCAA catabolism capacity, therefore an enzymatic issue, or more likely, various combinations of the first 3 options. BCAA levels may lower with fasting, eating less amino acid sources in the diet, therefore less high protein products, exhaustive exercise, and or increased BCKD enzymatic activity. We can also say that generally, elevated BCAA levels in the blood increase the risk of disease, and the ability to rapidly degrade BCAAs in the blood is associated with high aerobic capacity and low body fat. With all the data showing the dangerous outcomes of elevated BCAA levels in the blood, why are people still selling them as supplements? Well, for a long time, BCAAs have been shown to display protein-sparing effects. What I mean by this is that proteins are made of amino acids, and amino acids are used for many physiologic processes in the body. So, in times of need, protein can be broken down to harvest amino acids. BCAA supplementation is thought to prevent this by supplying your body with the amino acids it needs, thus preventing the breakdown of protein and accelerating the recovery process. However, the data remain mixed. Some show BCAA supplementation to suppress endogenous muscle protein breakdown and/or promote recovery, while others report that this does not protect against protein degradation. Some have even shown simple glucose intake after exercise to be more effective than BCAA supplementation to reduce catabolism after exercise. BCAAs do seem to promote anabolic signaling. Unfortunately, the signaling isn't always associated with increases in muscle mass. It's important to remember that signaling pathways are often involved in a number of diverse pathway branches with unique functions, so sometimes even something considered a growth signal may be differentially processed in the pathway and lead to something completely different and unique to what we generally think of as that pathway's function. Some groups also show that BCAA supplementation does not lead to insulin sensitivity in type 2 diabetes. Also, I should mention the data surrounding the claim that BCAA supplementation promotes performance during exercise is mixed. Many studies comparing carbohydrate, BCAA, and carbohydrate and BCAA drinks during exercise have found that carbs are more beneficial than BCAAs alone, and that BCAAs often do not increase performance over the placebo. I see a lot of BCAA energy supplements out there, but remember, the energy is not coming from the BCAAs. It usually comes from caffeine. Plus, if you remember the McArdle's disease patient findings, energy from BCAAs is not the best for athletic performance. To add to this, one group investigated the effects BCAA and glutamine supplementation may have on juvenile rowers and found only glutamine was able to reduce measures of fatigue. But again, the question arises, why are there BCAA supplements? The main variable here seems to be exercise, or rather that you can efficiently break down BCAAs. Basically, if you use what you put in, then all is fine. But when you put in too much and/or the system backs up, problems arise. So the main argument is you put BCAAs in, use them appropriately, and levels return to baseline in the body without causing harm to the individual. Another variable to consider is food intake. It does seem that those low in amino acid intake or on a caloric restriction benefit more from BCAA supplementation than those already consuming enough amino acids in their diet. This includes vegetarians, vegans, and those who exercise. As an interesting side note, BCAA supplementation has been used in hepatitis C patients to reduce oxidative stress in the liver, is associated with reduced liver carcinoma incidence, and prolongs the survival of liver cirrhosis patients. So BCAAs do seem to help in certain liver disease patient populations. Overall, I would say supplementing with BCAAs can be safe and dangerous. Just like glucose, too much is a problem and too little is a problem. BCAAs and other amino acids are important for our survival and function, but ingesting high amounts when already consuming enough protein from your diet or supplements could lead to elevated BCAA levels in the blood and drive disease. On the flip side, people low in amino acid intake, like some vegetarians and vegans, will benefit more than those already receiving enough via their diet. More than anything, it's important to note that supplements are marketed well, like Gatorade. In general, the developing infant and child require significantly more amino acid consumption than adults. As for the RDAs, or the recommended daily allowances, in 1989, the adult RDA for leucine was 14 milligrams per kilogram body weight, isoleucine 10, and valine 10. However, these values changed in 2002. Leucine was then 39, isoleucine 20, and valine 26. In 2006, these estimations changed again to 42 for leucine, 19 for isoleucine, and 24 for valine. When looking at the tolerable upper level data about the BCAAs, leucine, when taken at about 39 grams per day or 550 milligrams per kilogram per day, was found to be a health risk for adults and elderly men. I was unable to find the same level of data for both isoleucine and valine. I will also say I think it's important to note the signs of low amino acid intake. These include muscle weakness and small size, edema, skin abnormalities, thin or fragile hair, low serum albumin, and there may be hormonal imbalances as well. These are the references I've used for this presentation and the image URLs. Again, this is Dylan with BJJ/J Athletic Training. Thanks for watching, and I hope you enjoyed this week's data-driven nutrition section focusing on branched chain amino acids.